Okay, now let's talk about the worst microorganisms. It's not an opinion, it's a fact. Fungi. So we're not going to use taxonomical classification, but we're going to use structural classification of fungi. Fungi generally can be divided into molds and yeasts. So molds are multicellular fungi. There's still an argument among mycologists and biologists at large whether we can consider molds multicellular or we should consider them colonial, because multicellular organisms imply differentiation into some kind of tissues. I'm in the multicellular camp, okay, just because it's easier to formally distinguish between molds and yeasts. So these multicellular molds form long chains of cells called hyphae, which you all have seen mold, I suppose, right? So this fuzzy appearance of a mold is called mycelium, and each individual thread in the mycelium is a hyphae. Now, hyphae can be, I'm just going to say it, so okay. Hyphae can be vegetative, which means they collect nutrients, or reproductive. In some molds, individual cells in hyphae fuse, forming so-called non-septate hyphae. So if you look down the microscope, you see multiple nuclei, but cells are not separated from each other. Does that make sense? So septate versus non-septate. Now, what are strategies, reproductive strategies for molds? Molds can reproduce via hyphal segmentation, which exactly what it sounds like. Does that make sense? So when you find something in the back of the fridge that looks like it's about to start talking to you, you usually, you don't want to open it. If it's a mold and you open it, fragments of hyphae will just start flying around your kitchen. It's not dangerous for you, but it may increase the risk of fungal contamination for the food, and it just spoil the food. They can reproduce using sexual spores, and reproduction using sexual spores is kind of rare because sexual spores are formed under harsh environmental conditions. Okay? Most common way to reproduce for molds is asexual spores, which are pretty common. And fungal spores are all over the place. There was an estimation that average human being inhales about 40,000 fungal spores a day. Okay, which is okay. Now there are two common types of asexual spores. It can be sporangiospores. Sorry. And conidiospores. So in case of sporangiospores, you have a vessel, a structure. Sporangio literally means vessel of spores. So something like this. And the spores are inside, okay? And in case of conidiospores, you have individual spores produced at the end of the hyphae. Okay, those are two structural types that I want you to be able to distinguish. We're good? Okay, now, second type of fungi are yeast. These are unicellular fungi. Okay, now they reproduce by budding. That's first most common way. And budding is a glorified name for cell division, from mitosis. Does that make sense? So what you end up with, imagine that this is the fungal cell, my fists, okay? The yeast cell. The new cell buds out of it, and now you have two yeast cells. You following me? Two yeast cells. These two yeast cells, after budding, they can stay connected to each other. And each of the cells is an individual independent microorganism, but they just stay physically attached, which results in the formation of long chains of individual cells called pseudohyphae. Okay. Other two strategies for yeast reproduction include, just like with molds, asexual, common, and sexual, rare spores. And the requirements for the formation of both are similar. Okay, so sexual spores are formed under the harsh conditions and rare. Asexual spores form normally and are common. Any questions so far about two different types of fungi? Okay. Now let's say a few words about an interesting type of fungus, or fungi, which are called dimorphic fungi. We talked about them before. Dimorphic fungi grow as molds in nature, and when they get in apparently warmer human body, they grow as yeast. Does that make sense to you all? You good? Now I want to kind of highlight a very important common feature of all fungal infections. I'm going to put it here because we're going to talk about antifungals and then certain fungal types. So, listen. I'm not asking for that sensitive medical information, God forbid, but I'm just going to say, you probably know, or you experienced, you know someone with that condition, or you experienced conditions like thrush, or athlete's foot, or ringworm, or I don't know, vaginal candidiasis, stuff like that, you know, the yeast infection. You know someone, I'm 100% sure, or you had, like I had athlete's foot when I was a kid, okay? And surface infections with fungi, infections that run on the cutaneous membranes or outer mucous membranes, are pretty common. Does that make sense? I can almost bet that you do not know anyone who had systemic fungal infection, because human body is to one fungi. So fungal infections, as a general rule, are superficial in immunocompetent individuals, can become systemic in immunocompromised. Does that make sense? You actually, like, we're going to talk about some fungal infections now, and you will see that they affect, when we talk about systemic infections, that's going to be AIDS, immunosuppressed, AIDS, immunosuppressed, AIDS, immunosuppressed. Does that make sense? So, coccidioides immitis may sound really, really terrible, but you all are not at risk for coccidioidomycosis because you're not immunosuppressed. Are we clear? Got it? So just remember, fungal infections are systemic in immunocompromised and kind of superficial, so cutaneous or superficially on the mucous surfaces in immunocompetent. We good? Okay. Let's talk about antifungals. So what is the main issue with antifungal treatment? Fungi, are they eukaryotes or prokaryotes? Do they have a nucleus? I actually said it today already. Eukaryotes, prokaryotes? Eukaryotes, yeah, they're eukaryotes, just as we are. So targeting specific processes becomes a little bit challenging, right? So they also have eukaryotic cells. So there are 4 classes/types of antifungals that I want you to be aware of. First group is polyenes, which target, and I say, I put this cross over here. Doesn't mean that they inhibit, they basically pierce the membrane. They target the membrane of the fungal cell. As you can imagine, polyenes are pretty toxic. Amphotericin B is the great example. It's a very, very bad, it's a great drug for treating systemic fungal infections, but it has a lot of side effects. Okay. Then we have griseofulvin. It's a decent oral drug that treats, for instance, cutaneous mycosis, such as ringworm, and it inhibits the formation of microtubules. Flucytosine inhibits DNA synthesis and finally azoles or azole drugs like ketoconazole, fluconazole, myconazole, they inhibit ergosterol synthesis. And ergosterol, this molecule, is the key component of the fungal cell membrane, which is not present in a human cell membrane. So selective inhibition of ergosterol synthesis inhibits fungal growth, and that's it. Does that make sense? So these are 4 types/classes of antifungals I want you to know. Okay, we're good? Now let's talk about some diseases. First we're going to chat about, I call them cutaneous mycosis, which is not entirely correct. So what you need to know, you need to be able to recognize names basically. 4 genera of fungi that cause purely cutaneous mycosis are, now look at the names, epidermophyton. So phyton refers to some kind of growth, like a plant of sorts. Epidermo, skin. Trichophyton. Tricho refers to hair, okay? And you have a couple more, it's Microspora and Malassezia. All those are genera of fungi. They all cause cutaneous mycosis. Things like now this may come as a new term for you, tenias, right? Tinea. The word tinea as a medical term means a worm, okay? Now, those are fungi. They have nothing to do with worms. Am I clear? For some reason, though, you're familiar with the term ringworm, right? There's nothing to do with worms. This is where tinea is coming from, depending on the location, it can, for instance, if it's a human body, like a trunk, it's going to be Tinea corporis. If it's on the head, it's Tinea capitis. If it's on the chin, often it develops when men shave and damage the skin. Tinea barbaris, okay? If it's in the inguinal area, in the groin area, Tinea cruris. If it's on the feet, it's Tinea pedis.
Do we need to know all those?
Not really, just common sense, just recognize. Again, here's the difference. You think about this. If I ask, like, Tinea capitis, think capita, head, on the head, right? Those questions are not trippy at all. And I'm not asking you to provide me with a full list. Okay, so does that make sense so far? Now a couple more. One, cutaneous, causative agent of cutaneous aspergillus. Secondary mycosis. What does that mean? We're going to go back to aspergillus as the causative agent of respiratory infections, but let me tell you this. Secondary mycosis means that there is somewhere a primary infection with aspergillus. You with me so far? And it traveled through the bloodstream and basically popped through the skin somewhere in the periphery and caused mucosal lesion somewhere there. So basically what I'm trying to tell you is that if somebody develops cutaneous aspergillosis, it's the least of that person's problems. This person has something much more severe going on. And finally, I'm going to put mucous mycosis, Candida albicans. Now, Candida albicans not necessarily is present only on mucosa, but it's very well known for its mucosal infections. So it's a yeast infection. Thrush. That makes sense? Candida can also infect nails. It's a pretty common nail pathogen. So far we good? So next group will include, and I want to make it clear again, when we talk about fungal infections, we're talking about main portal of entry being lungs. Those fungi are inhaled. Does that make sense? That's absolutely not ingested, inhaled. Okay? So respiratory mycosis, we've got Pneumocystis jirovecii. It was really embarrassing yesterday when we were talking about it with a different class, and I named it Pneumocystis carini, which is the old name, and this is why I say that taxonomists have their own very well classified and constantly renamed place in hell. I don't really understand why they took Pneumocystis carini and renamed it jirovecii. It could have just stayed carini. Causes pneumonia. That makes sense? Now, what's interesting about this particular pathogen? This pneumonia, also known as PCP, Pneumocystis, old name carini pneumonia, is the hallmark infection for AIDS patients. Does that make sense so far? So basically, we're kind of creating a clinical picture of a person who suffers from AIDS. It's going to be fungal pneumonia. It's going to be skin cancer, Kaposi sarcoma, herpes virus. We're not talking about those protozoa, but it can be protozoan diarrhea. So it's kind of a complex condition which, even from symptoms, can be sometimes diagnosed. Different species of blastomyces. Also pneumonia. And Aspergillus. You know Aspergillus as the black mold, okay? And it causes a very interesting form of lung infection. Aspergillus doesn't readily establish the systemic disease, but it's very good at forming what's called hyphal balls. Okay? So can you imagine a mole growing on your produce, for instance? Imagine this. Imagine that same mold grows in your lungs. Exactly the same form, forming a little ball of mold within the lungs. Okay? And then it can take off and form secondary cutaneous mycosis somewhere else. That makes sense? What I want you to understand very, very clearly, you've got to be immunosuppressed to have this. It's not normal, like it's not going to happen to a person who is immunocompetent. Does that make sense to you all? Okay. So, what do we have next? We have neurotropic. Mycosis. There are just two that I want you to know. Cryptococcus neoformans. Now, this microorganism is a typical dimorphic fungus. It grows as the yeast in humans and causes meningitis. It's a hallmark disease for AIDS patients. One more stroke for the clinical picture. And it has a distinguishable, on the microscope, it has a very distinguishable virulence factor. Cryptococcal cells have a very, very thick capsule which protects them effectively from phagocytosis. So now, to the clinical picture of AIDS, we just added another person, with low-grade fever, stiff neck, headaches from cryptococcal meningitis. Good so far? Now, Cryptococcus neoformans is, another one, I'm going to put them together. Coccidioides immitis, also a causative agent of meningitis. And coccidioides is extremely contagious. Now, both of them are transmitted by the inhalation of spores that are in bats and bird species. So those are typical environmental fungi. Does that make sense so far?
Like they're like in their bodies or their poop?
In the poop. In the poop. So when it's aerosolized, people inhale it. I know that it may sound unprofessional to bring up House MD as the medical example, but they actually had, I think they had an episode about a patient with AIDS who had cryptococcal meningitis, and patient acquired it because the patient was, the pigeons, the people, he was breeding pigeons or feeding them, and they were like shitting all over the place and basically inhaled it stuff. That make sense? Again, fungal infections are rare. On the other hand, I listen to clinical updates on COVID situation in the United States, and it's a guy who is a chief infectious disease officer for a huge healthcare provider in New York, and he says that they see in people who went and ventilator and then were, relieved from ventilator and they get better. But very common secondary infection is fungal pneumonia. He never specified what kind of, but fungal pneumonia is because people who are off ventilator, they're weak, they're immunosuppressed, immunocompromised, so no wonder. Does that make sense?